All right, welcome. My name is Sonia Berthasell. I'm a PhD student at University of Maine and one of the coordinators for this USDA Northeast Climate Hub GradCAP program. GradCAP is a virtual consortium of master's and doctoral students who are working on climate adaptation in agriculture, aquaculture, and forestry. We're glad you've tuned in to today's webinar on soils and plant growth, which will highlight the work of 3 of our GradCAP scholars. 2 quick housekeeping announcements before we begin. This webinar is being recorded for subsequent viewing, and please mute your computer speakers and call in via phone to ensure good audio quality. You can also, if you choose, mute the sound on your phone so that you can hear us, but we can't hear you if you want to listen in peace. With that, it's my pleasure to announce your speakers. Brooke Eastman is a PhD student in biology at West Virginia University and will present a talk entitled An Appalachian Breakdown, Carbon Storage in a Temperate Deciduous Forest Responds to Over 28 Years of Nitrogen Additions. After that, we'll have Sarah Mills, who's a PhD student in horticulture at West Virginia University, and will present on blueberries and bees under increasing temperatures. And Bill Errickson, who's a PhD student in plant biology at Rutgers University, and will present on improving abiotic stress tolerance in crops with Rhizobacteria. After all 3 presentations, we'll have time for joint question and answer at the end, so please hold verbal questions until then. You may type questions in the chat box throughout the presentations, which we'll then answer either via chat or at the end. And with those few announcements, I'll turn it over to Brooke Eastman.
Hello, thanks to everyone here listening today. I am going to be using a temperate forest in West Virginia as a case study to explore the long-term effects of nitrogen deposition on forest productivity, especially carbon and nutrient storage in the soil. To start, I just want to highlight the importance of forests and healthy soils and the subsequent ecosystem services they provide. Soil formation, nutrient cycling, and primary production are ecosystem processes that influence the magnitude and quality of the other categories of ecosystem services, which more directly benefit us humans. And I'm interested in how these ecosystem or supporting services influence the regulating services of climate and carbon sequestration. And I also want to briefly highlight the forests in our region and show some studies that have found the Northeast U.S. to be relatively important in terms of potential productivity and carbon sequestration. This map shows estimates of albedo, or reflectance, of the forest canopy back, reflecting sunlight back out of the atmosphere. And researchers from the study have also found higher nitrogen concentrations of the canopy tissue, as well as increased potential photosynthesis or productivity in these forests. And so I've circled here that it's evident that West Virginia and the Central Appalachian Mountains, as well as the Northeast U.S. forests, are have strong potential for forest productivity and high albedo. Additionally, the 3rd National Climate Assessment found some of the highest rates of forest productivity in West Virginia, with still relatively high rates in the rest of the Northeast compared to Western U.S., further emphasizing the importance of the forests of our region. What this map doesn't show is soil carbon storage or sequestration. It's much easier to measure above ground biomass than it is to measure soil properties. But soils actually contain the largest pool of terrestrial carbon, as well as serve as a kind of foundation for above ground production and the provision of many ecosystem services we considered earlier. And if we think about the global carbon cycle, we can see here that soils contain more than land plants and the atmosphere combined. And also the flux of carbon from the soil to the atmosphere is over 6 times that of human activities or human emissions. So you might imagine that a small change in the flux of carbon from the soil to the atmosphere can have a pretty strong influence on the land-atmosphere exchange of carbon and ultimately the trend of accumulating carbon in the atmosphere. And any shift in the pool of soil carbon reflects a balance between carbon inputs to the soil as well as carbon outputs to the soil. And these inputs and outputs can be affected by many different factors, from primary productivity and carbon allocation in trees to soil enzyme kinetics and microbial communities. And all of these mechanisms are really tightly linked to the nitrogen status of a forest, because nitrogen can limit tree growth as well as influence soil microbial activity. So we can't ignore the nitrogen cycle when considering forest ecosystem functioning. And forests in the northeast U.S. have historically received really high levels of nitrogen deposition from acid rain. And this map shows nitrogen deposition levels at pre-industrial rates in the top map, as well as in the 1990s when acid rain rates were at their peak. And then finally, predictions for 2050 of nitrogen deposition, which would probably be most likely due to agricultural inputs of synthetic nitrogen fertilizer. And while we don't consider nitrogen deposition a part of climate change per se, it is intrinsically linked to climate change because nitrogen availability influences a forest's response and adaptability to climate change. And so as temperatures and atmospheric carbon dioxide levels rise, the ability for forests to further sequester carbon and increase their productivity under those conditions will depend partly on nitrogen availability. And a meta-analysis of forest nitrogen addition experiments found several results, and I'll just highlight a few here for you. They found no change in litter fall inputs with nitrogen addition, so these carbon inputs to the soil that I talked about. But they did see a diminishing in soil respiration or losses of carbon from the soil. And with that, they've observed an increase in the soil carbon pool. So if the inputs remain the same and the losses are decreased, then one would expect an increase in the soil carbon pool. I additionally want to highlight the about 36% increase in total biomass, which is quite striking. But this is a little bit more variable of an observation. And I also want to note that a lot of these studies of nitrogen additions in forests have been long-term or have not been as long-term, or maybe have not been done in natural ecosystems. And so I'm kind of interested in wondering if these responses would hold true over long-term or chronic nitrogen addition. And so to test that, I am utilizing the Perennial experimental forest whole watershed acidification experiment. And this experiment consists of a paired watershed. So the fertilized watershed 3 receives about 35 kilograms of nitrogen per hectare each year, and this fertilization started in 1989. And then an adjacent watershed 7, which is a similar age, serves as a comparative reference watershed. And so this paired watershed study is allowing me to explore questions like, is the increase in biomass production just an initial stimulation, or is it long-lasting? Do soils continue to sequester carbon for decades after elevated nitrogen input, as well as explore more in-depth mechanisms for the forest response to nitrogen deposition. And just to give a little bit more background of the site of the Furno and some environmental trends, I've shown here the history of nitrogen deposition at the Furno. On the left, so you can see that after the Industrial Revolution, nitrogen deposition increased quite rapidly, followed by a decline after the 1990 Clean Air Act amendment, back down to about 1950s levels of nitrogen deposition. And so this figure highlights both the importance of long-term data collection as well as the effectiveness of policy in improving environmental conditions. However, on the right, you can see that these nitrogen depositions have added nearly 300 kilograms of nitrogen per hectare to the Furno since the 1900s, early 1900s. I've also included some data from NOAA on mean annual temperature on the left, and then annual precipitation on the right. And the blue trend lines show not much of a trend over this period, which was from 1930 to present day. But while we don't see a trend over this time period, this long-term data set is really valuable and may suggest otherwise in the future. But from this slide, you can kind of see that nitrogen deposition trends are really clear and likely influencing the forest. And so part of my exploration of the forest response to nitrogen additions has been to construct a carbon budget for these watersheds. And I just want to highlight a few key components of this budget, starting with above ground biomass. And so this figure here on the right shows that there's been a stimulation in basal area increment or tree growth in the fertilized watershed, which is shown in yellow, compared to the reference watershed shown in blue. And I'll use these same colors for the rest of the presentation. But if you look across different time periods between measurements, tree measurements, one might initially suspect that there's just an initial stimulation of tree growth from 1990 to 1996, followed by perhaps a decline in tree growth following that. But our most recent measurements showed, again, that the fertilized watershed appears to be growing faster 20 to 30 years after the fertilization began. And then in terms of the below ground or the soil carbon pools, I've been looking more closely at inputs and outputs of carbon to the soil. So starting with the inputs, or leaf litter fall, and in this figure we don't observe any change in leaf litter inputs to the soil. The trend line, the blue trend line is a little bit hard to see, but it's kind of obscured by the yellow trend line here. But basically both watersheds are increasing slightly their carbon inputs to the soil through leaf litter, but at the same rate over time. And then in contrast, we see slower litter decay rates as well as reduced soil enzyme activity in the fertilized watershed, suggesting potentially fewer losses of carbon from the soil. And so if above ground inputs are equal and the breakdown of carbon in the soil is lower, then we would expect less losses of carbon through soil respiration. Additionally, we see lower standing root stock in the fertilized watershed, but faster fine root turnover. So there's less roots overall, but these roots are dying and producing new roots more quickly. And so again, with lower standing root stock, we might observe less respiration from these roots coming out in total soil respiration measurements. And so to test that, I measured soil respiration at 40 sites in each watershed for 2 years and found that indeed the fertilized watershed had less carbon losses from soil respiration than the reference watershed. And this difference from this figure over time appears to be quite small, but when scanned to annual losses of carbon, we do see an 11 to 16% decrease in carbon losses with nitrogen addition. And if you remember the global carbon cycle presented earlier, even these small changes in soil respiration can really influence the global carbon balance between the land and the atmosphere. And so what does this Appalachian breakdown mean when pieced back together? And I really like this analogy of the bathtub with a faucet and a drain to represent sinks or sources. And in the case of the fertilized watershed at the Perennial, the inputs of carbon to the forest soil or water from the faucet into the tub are maintaining the same rate as the reference watershed, but the drain seems to be a bit clogged. So there are less losses of carbon from the fertilized watershed as we observe reduced microbial and fine root respiration. Also, losses of dissolved organic carbon in streams in the two watersheds were equal in recent years, so we do not think that carbon is being lost through leaching. Therefore, unless carbon is being lost through some other process that we haven't measured, then the soil carbon stock is likely increasing, or the bathtub is filling up. However, we have not been able to detect a quantitative change in the soil carbon pool despite these diminished losses from the soil. And we think that this is likely because the carbon pool in the soil is so large and the change is so small that it's hard to detect this change in the scale of about 30 years. But what we have observed is a shift in the chemical nature of the soil organic matter. So the organic matter in the fertilized watershed has a greater carbon to nitrogen ratio. This may mean that the organic matter is more plant-like and less microbial-like, which would be supported by our observations of slower litter decomposition or less enzyme activity. And this may have implications for further carbon storage and nutrient cycling in the future under different environmental conditions by influencing its resistance or susceptibility to decomposition. So again, if we consider the global carbon cycle again, we can see how even a small change in soil respiration can have a significant effect on the global carbon cycle. And this change of 11 to 16% is one that has been observed both in short-term nitrogen addition studies as well as in this study of the Furneaux after almost 30 years of nitrogen addition. And so the interactions between carbon and nitrogen cycles in temperate forests are really important to consider for future management, as the nitrogen status and the stoichiometry, or the carbon to nitrogen ratios of organic matter and microbes, are likely to influence the forest response to changes in climate. And so, in conclusion, we've observed enhanced overall carbon storage, partly through reduced losses of carbon from the ecosystem after almost 30 years of nitrogen additions at the Furneaux experimental forest. These were quite consistent with other studies, other long-term nitrogen addition studies. But we also recognize the need to better understand how this added carbon in the soil, or this changed nature of the carbon in the soil, will respond to changing climate. And I also want to argue again that nitrogen also must be considered in order to effectively predict how ecosystems may respond to a changing climate or changing management of forest ecosystems, as well as highlight the importance of long-term data measurement for understanding these ecological processes, which with that then I can go ahead and thank the Northern Research Station in Parsons, West Virginia, for their long-term monitoring and research at the Furneaux, as well as everybody else listed here who have assisted with this project. Thanks so much.
Thank you, Brooke. And a reminder, if you have questions for Brooke, hold on to them and we'll do joint question and answers at the end of all of our presentations today. You can also type questions into the chat box as we go, and maybe Brooke can respond to some that way during our next presentation. Next up, we'll be hearing from Sarah Mills, and bear with me for 1 second while I switch over to Sarah's presentation. Take it away, Sarah.
Hi, everyone. Thanks for attending this webinar. As a quick reminder, I'm Sarah Mills, and I'm a PhD student at West Virginia University. And I'll be talking to you today about a grant that's focusing on the effects of temperature on blueberry and their pollinators. So blueberries are an economically significant crop. And just last year, they were valued at almost $840 million. And 82% of that is actually utilized fresh. And here you can see that this, here you can see, well, in the figure, you can see that there's a dramatic increase in the production of blueberries over the last 25 years. Blueberries are a perennial crop native to North America, and they were actually first cultivated right here in the Northeast about 100 years ago. And while now they're grown worldwide, 90% of the world's blueberry production still comes from North America. Blueberry popularity has increased dramatically, probably due to their great taste, but also to the health benefits. So blueberries are an icon for a nutritious lifestyle. They're a great source of vitamin C and manganese. But blueberries are also high in antioxidants, but they're not limited to human consumption. Blueberries are currently found in over 4,000 products, some of them being cosmetics and also pet food. Flower initiation begins in the fall. And so currently that's what blueberries are working on, is setting flower and leaf buds for the following year. As they prepare for winter dormancy, they'll shed their leaves. And winter, cold temperatures are required in order to meet their chilling requirements. The transition from winter to spring is dependent upon temperature. And early spring flowering is common for blueberry. And then finally in summer, you'll have ripe fruit ready to harvest. But these fruit production depends on flowers. In the first picture, you can see full bloom flowers, which are mostly open and available for pollinators. In the next slide, you'll see petal fall, which is the natural senescence of the corolla. And if you see at the base of the white flower is actually the ovary, which then swells to become the fruit. And then the fruit swells in size and then develops pigment, and then is the recognizable ripe blueberry fruit. But as I said, blueberries are highly dependent upon pollinators for adequate fruit production. Here you can see this picture is displaying fruit that was harvested on the same day. While some flowers were bagged to exclude pollinators, others remained open. And you can see that while some fruit set is possible, because blueberries are self-compatible, opening to pollinators and cross-pollination is advantageous. And in some cultivars, cross-pollination can increase yield by 10 to 20%. But the growth of blueberries and the activity of pollinators is dependent upon their environment, particularly on temperature. So here you can see that the pre-industrial baseline is the temperature prior to the Industrial Revolution. But you can see that the last several years, the temperature has dramatically increased above that baseline. And this temperature increase is only expected to continue. Models looking at the national proposals, these are expectations in the temperature rise by 2100 if accounting for pledges to reduce carbon emissions. In the business as usual model, they don't account for any reductions in carbon emissions. And so we can see that by 2100, we can expect a 3 to 4 degree increase in average temperatures globally. So the objective of this project was to look at the effects on temperature, look at the temperature effects on blueberry and the emergence of bees. Since blueberries are dependent upon pollinators, the interaction between these two organisms is really important and can be disrupted by changes in our environment. So this experiment utilized 5 different temperatures. As the horticulturalist, I contributed to the blueberry portion, and my collaborators allowed me to share the bee data. And so we studied 2 different cultivars of highbush blueberry, Blu-ray and Jersey. And these 2 cultivars were selected based off differences in their fruiting period. First, we can see that the number of flowers produced can be affected by temperature. Here we can see that in Morgantown, West Virginia, 18 degrees Celsius is a common temperature of what we would expect to see in April, which is when blueberries would typically be flowering. As you can see here, a slight increase in temperature may actually be beneficial for the development of flowers for blueberry. But too much of a temperature increase is going to be detrimental. As you can see, if we shift from 24 degrees to 28 degrees in Blu-ray, which is the first cultivar, you can see about a 70% decrease in the numbers of flowers produced. While in Jersey, the other cultivar, you can see about a 35% decrease. The rate of development is defined as the amount of time it takes to progress from early pink, which is that purple box in the top corner, where we have closed flowers that are individually identifiable, and then the rate it takes from early pink to full bloom, which is when flowers are completely open. First, we can notice that the shift in temperature of increasing from 18 to 20, so just 2 degrees, we can see that the rate of development, or how quickly that's progressing, is 5 days faster. And while we see that for blue-ray, almost every temperature shift from 18 to 28 resulted in an acceleration of development, we only see one temperature shift for Jersey, and that's between 24 and 28. Bee emergence follows fairly similar trends to the plants. Here, the first figure on the top is showing like a typical nest of what a solitary bee nest would look like. And you can see that bees are currently in their larval stage. Here in this figure, you can see the time it takes for bees to emerge or first exit the nest actually is much longer in colder temperatures. And comparing 18 to 30, we can see that bees took twice as long to emerge at colder temperatures than warmer. Here, we're integrating the flower and bee data. You can see we compared the full bloom stage with the emergence of adult bees. In this figure, that light green, which is the dominant color across most of the Northeast, shows that bees will emerge 10 to 20 days prior to flowers. And so if we take a closer look at Jersey, or New Jersey, the state as an example, we can see that the southern portion is in yellow, which is 20 to 30 days. And then that separation is only expected to increase as we continue into the future. We can see the addition of warmer colors, increasing a longer separation between flowers and their pollinators. So an average bee only lives for about 6 weeks. And so if we look at 30 days separation, we're looking about 70% of its lifespan waiting for a flower, waiting for a food source. And so this is problematic. With the separation in time between the bees and their flowers, bees are likely to either find another food source or risk starvation. Another thing to consider is that individual flowers of blueberry are only open for about 5 to 7 days and are ideally pollinated within 3. And so separation between even 3 days between flowers and their pollinators is significant and reduces yield and quality. So overall, temperatures are likely to reduce the flower and fruit production simply because each fruit comes from a flower. So any reductions in flower number will reduce production of fruit. We also saw accelerated rates of development and rates of emergence for the bees. And this is going to cause an issue as if flowers are going to be, or blueberries are going to be flowering earlier in the spring, it makes them susceptible to late frosts and frost damage. It also shrinks that window even further between blueberries and their pollinators. And this will likely result in an asynchrony between the plants and pollinators, and likely we'll have to manage bee emergence in order to sync the two. However, we did see differences in cultivar response. This indicates that there is genetic diversity within blueberries currently on commercially available. And so breeding programs or cultivar selection can be used to improve blueberry plantings, as blueberries can take 5 to 7 years to reach maturity and can often be planted for 30 or more years. So long-term investments. Finally, I'd like to acknowledge my lab and collaborators for their contributions to this project. I'd like to thank the GradCAP program and the Northeast Climate Hub for hosting us. And then finally, my funding source with the USDA.
Thank you so much, Sarah. Our third presenter today will be Bill Errickson from Rutgers University. And again, please bear with me for just one moment while I switch over to Bill's presentation. Again, you can feel free to send immediate questions for Sarah into the chat box if you'd like. Go ahead, Bill. Bill, are you there?
Yes.
Oh, great.
Looks like the presentation is still loading.
Oh, really? All right. Give it just one more moment while it comes up. It seems to be sharing on my computer.
Does anyone else not see it?
I don't see it either. Oh, it just popped up.
Okay.
I don't see it
There we
either.
go.
Apparently it's just slow. Give it a second.
Yep. Thanks for your patience with the ways of the internet.
We have very skinny lines coming out of Maine.
Mm-hmm.
Everybody have it on their screen?
I think so.
All right.
Go ahead.
Great. Hi, everyone. This is Bill Errickson. I'm a second-year PhD student at Rutgers University in the Department of Plant Biology. And this webinar is going to focus on some of my research on improving abiotic stress tolerance in crops using Rhizobacteria. As the climate changes, we continue to see an increase in the severity of abiotic plant stressors such as heat, drought, and salinity. These stressors have various physiological and morphological impacts on plants, ultimately leading to crop losses and decreased yields. Growers need effective and sustainable methods to mitigate abiotic stress and reduce crop losses as part of their climate adaptation strategy. Plant growth promoting Rhizobacteria, or PGPRs, can reduce the impacts of abiotic plant stressors and provide growers with effective options to help build resilience into their cropping systems. Three major impacts of climate change include increases in heat, drought, and salinity stress on crops. These stressors are major factors limiting plant growth and productivity. The years 2012, 2016, and 2017 were the three hottest on record in the U.S., with average annual temperatures higher than any other year since temperature data was first collected. In 2017, every state in the U.S. had a warmer than average annual temperature. In the Northeast, growing seasons are becoming longer. However, summer heat stress days are also increasing, in which the temperature is exceeding the natural optimum range for many regionally adapted plant species. This stress on plants is further compounded by drought stress, which often occurs in conjunction with heat stress. Severe drought across the country in 2012 caused the USDA to declare a natural disaster for over 2,200 counties, representing 71% of the United States. While the Northeast is not predicted to incur the devastating long-term water shortages of other regions, short-term summer drought conditions are expected to increase. Salt-affected soils account for approximately 50% of total irrigated land in the world. The annual economic loss in crop production due to salinity stress is severe, currently totaling several $billions. Salinity stress is continuously expanding due to the increasing use of effluent water for irrigation, saltwater intrusion in coastal areas, and high evaporative demand in areas with salt-affected soils. So, When a plant perceives abiotic stress, it responds by producing a hormone called ethylene. First, the plant produces a small peak of ethylene, which triggers the expression of stress response genes. This causes the plant to produce a second, larger peak of ethylene, which results in premature leaf senescence, reduced growth, and ultimately lower yields. Plants synthesize ethylene from another naturally present compound called ACC, which is amino cyclopropane carboxylic acid. Some rhizobacteria have plant growth promoting properties, and one of these properties involves the ability of the bacteria to break down ACC using an enzyme called ACC deaminase. The ACC is converted into ammonia, which the bacteria uses as a nitrogen source. These bacteria can form a symbiotic association with the roots of many plants to reduce the production of stress ethylene and encourage growth in the presence of abiotic stress. Previous research in our department involved the collection of rhizobacteria from the roots of native grasses in the New Jersey pine barrens. The pine barrens have sandy, acidic soils with low fertility and frequent drought conditions, representing a native habitat where plants and their rhizobacteria symbionts have adapted to abiotic stress. We have isolated and identified approximately 120 unique strains in this collection of rhizobacteria, with the majority coming from the genus Burkholderia. These rhizobacteria are predominantly endophytes, meaning that they live inside the root tissue of the plant as opposed to living in the soil. We screen each strain in the collection for various growth-promoting properties, including ACC deaminase activity and the production of plant growth-promoting hormones, such as IAA, which is an auxin involved in root development, and gibberellic acid, which stimulates growth and cell elongation in roots and stems. When we have identified a strain that has significant growth-promoting properties, we conduct growth chamber inoculation trials. The bacteria are cultured in a liquid medium, then the cells are extracted and resuspended in water to make an inoculum. This is watered in as a soil drench at the 2 to 3 leaf stage of seedling development. We then subject the plants to various abiotic stresses for a period of time and measure the physiological and morphological responses. This study evaluated salinity stress on perennial ryegrass, in which we observed increased shoot and root biomass in the inoculated plants after 20 days of salinity stress. On the left, we have non-inoculated plants, and on the right, we have plants inoculated with RU1, which is a newly patented PGPR strain discovered at Rutgers. We observed a similar response in tomato, which also demonstrated increased shoot and root biomass with inoculation under both salinity stress and non-stress ambient control growth conditions. On the left, we have non-inoculated and inoculated plants under optimum conditions, and on the right, we have non-inoculated and inoculated plants after 21 days of salinity stress. Corn demonstrated increased root biomass and root length with bacterial inoculation under salinity stress and optimum growth conditions. On the left, we have non-inoculated versus inoculated plants under optimum conditions. And on the right, we have non-inoculated versus inoculated plants after 14 days of salinity stress. We've also evaluated the effects of inoculation with several different strains of rhizobacteria on heat and drought stress in creeping bentgrass. Once the plants were inoculated, they were subjected to either 21 days of drought stress or 35 days of heat stress. The physiological response included increased chlorophyll content and photochemical efficiency with inoculation, representing an increased ability for the plant to convert sunlight into glucose and potentially into biomass. The photo is showing how we measure photochemical efficiency. The white clips are placed onto the leaves and they are dark adapted. We then use a machine to shine a light on the leaves and measure the fluorescence. Morphologically, we observed increased root diameter and root surface area with inoculation in both the heat and drought stress scenarios. On the left, we have non-inoculated plants versus inoculated plants under heat stress. And on the right, we have non-inoculated versus inoculated plants under drought stress conditions. The implications of this work suggest that plant growth promoting Rhizobacteria can provide growers with effective and sustainable methods to mitigate abiotic stress in crops, including increased productivity, reduced environmental impacts, and increased root carbon sequestration. Future work includes further investigation into both the applied and mechanistic aspects of this research. We're continuing to screen additional strains of bacteria and exploring other growth-promoting properties, such as nitrogen fixation and cytokinin production. Developing the Rhizobacteria into biofertilizers or inoculants that can be field-applied represents another challenge, as native microorganisms already present in the soil can cause competition with the PGPRs. While our early work has been focusing on the seedling stage, we also plan to evaluate the effects of inoculation on crop yields. We also want to understand the mechanisms involved of the interactions, including changes in gene expression, proteins, and metabolites. I would like to thank my advisor, Dr. Bingru Wong, one of our collaborators at Rutgers, Dr. Ning Zhang and her lab, the Rutgers University Department of Plant Biology and Pathology, the USDA Climate Hub, and some of our funding sources, including USDA SARE and the O.J. Noer Foundation. Thank you very much.
Thank you, Bill, and thanks to all three of our presenters. So in just a moment, we will now move to a Q&A, but before that, I have one quick announcement. The next webinar in our series is shown here with a blue arrow. The theme will be changing ecosystems, and you'll hear 3 more presentations from GradCAP Scholars coming up on October 18th at noon. So please consider tuning in for that. And details about that and all of our webinars can be found by going to our GradCAP website, which is the link at the bottom of the slide that you see before you here. Also, very soon, recordings of past webinars will be housed on this website as well. So we're glad you tuned in today and hope to see you again. Now, please, if you've got questions for any of today's presenters, feel free to launch in with those, either via phone or in the chat box. Looks like Cara is starting us off with a question for Bill. Were different types of soils compared, or are there plans to look at this? For example, sandy soils versus loam soils. Bill, you can either type your reply or go ahead and answer that verbally if you'd like.
I started typing, but I'll answer verbally. So currently in the growth chambers, we're using calcined clay, which is a medium that's almost like it's almost the texture of a coarse sand. And so this allows us to sort of have a clean slate for inoculation and not have, other organisms present. I think once we get to the field adaptation stage, we might look at different common soils and probably start by autoclaving them to sort of start with a clean slate and do soil trials within the growth chambers and greenhouse, and then try to move that out to the field.
Cool.
And
you want to address Cara's second question there as well?
Yes, that's another aspect that we're working with is sort of optimizing the bacteria, plant, and stress interaction. So not only do certain strains of PGPRs work with certain types of crops, but also with certain stresses. So one strain might work better in heat stress, another in drought stress, and another in salinity stress. And so we're trying to optimize those interactions as well.
Great, thanks. Sarah, Ivan's typed in a question. How do blueberry growers manage to protect from late frost?
Hey, so late frost can just kind of occur, and so sometimes they just kind of deal with it. Depending on the stage of the bud, the blueberry flowers are actually fairly cold tolerant, but like the most vulnerable stage is that petal fall, is when we're trying to transition from an open flower into the fruit, and that can only handle temperatures to about 32. And so, but below that, the bud can actually handle like mid-20s Fahrenheit. So frost damage can still be detrimental, but not, there is cold tolerance of there. Sorry, there's a new question coming in.
Oh, that's quite all right. You can finish addressing that one, and then we'll move on to the next.
Okay, sorry. I was getting distracted. But generally, they can either incorporate some like heaters into the fields or into their growing environments. They can apply some of the sprinkler systems to provide some protection over to the flowers, things like that.
Great, thanks. And then you can go ahead and answer Kara's question about how the 2 blueberry cultivars were selected.
So there's about 4 different distinct stages of like categories of fruit ripening for blueberry. So there are cultivars that will be early season versus mid-season, late season, things like that. And the 2 cultivars were selected based off of mid-season and a late season. And also Jersey, there's been previous studies that show that Jersey is actually an heirloom and is fairly heat tolerant.
Cool, thanks. Next, I see a question for Brooke. Are there differences in hydrologic yield between the 2 watersheds, the W3 and W7 watersheds in terms of tree growth differences? Oh, due to tree growth differences. I read that question wrong. You can read it correctly and answer it at your leisure.
It's okay. I haven't looked at that, actually. That's a really good question. I think it would be hard possibly to attribute that maybe to tree growth differences, just because I think there are different capacities of the soils to hold moisture as well. I noticed that in watershed, the fertilized watershed 3 has typically higher soil moisture than watershed 7. So perhaps that might also contribute to any difference if there is in the hydrologic yield. That's a great question, something I should definitely look into.
Cool. Sarah, there's another question for you here. Do you know anything about whether blueberries are more or less sensitive to warming increases than other kinds of small fruits?
I would expect so. I would probably put blueberries more in the classification of the perennial crop, so compare them more to their sensitivities like apples than I would to other small fruits. Other small fruits actually don't produce buds prior to overwintering, so they're producing within the same growing season, and so they're not even experiencing that early spring flowering. They're normally flowering in like mid to late summer, and so they're not going to be as exposed to that cold temperature, and so they're going to have like less stress there.
Great, thanks. Then Bill, another question for you. Are there other management techniques used that involve the addition of biological inoculants?
In terms of other functions of the organisms, if I'm understanding that question correctly, there's, definitely biological inoculants that can be used for pathogen control, phosphorus solubilization, nitrogen fixation, so, Rhizobium being one of the most common ones we think of.
Yeah, which is applied to our legume crops often.
Yeah.
Okay. Yep, that was the question.
Awesome. Thanks for the questions. Anyone else have questions to throw out into the world? If not, I'm sure you can email us or email our presenters after the fact if you look them up online. And we hope you'll tune in to our next webinar on October 18th.
Any questions from anyone?
Any final questions? All right.
Okay, great. Thank you very much.
Thank you so much.
Presentations were excellent.
Thank you.
Thank you very
much.
I'm going to stop recording.